Hey guys, welcome back to the channel. So in this video, we are going to study about agriculture microbiology. So let's get started. So previously we talked about Kingdom Monera and I just gave an overview about what is all about Kingdom Monera. So we talked about some of the organisms and bacterial species and some of its features. So let's get deep into this and study and get to know more about this. So Kingdom Monera, the nutrition is can be any or can be of any sort, which can be autotrophic, heterotrophic, saprophytes, parasites, symbiotic, or commensalism. So autotrophs can prepare their own food, heterotrophs depend on others for food or basically depend on autotrophs for food. Saprophytes feed on dead and decaying matter for food. Parasites live on host cells for the survival. Symbiotic ones can be in a mutual relationship with other organisms where both of them get benefited and the commensalism is opposite of symbiosis, which is one organism is benefited and other is affected. So these are some of the basic things that I've already covered in some of my previous videos, so you may check them out. So these were the nutrition types of Kingdom Monera. So with this, so talking about some of the chemosynthetic autotrophic bacteria that comes under Kingdom Monera. So the chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia that uses energy for their ATP production. Also, they play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. So chemosynthetic autotrophic bacteria are none other than these are a type of autotrophic bacteria which basically utilizes inorganic substances, which are mainly nitrates and nitrites, for ATP generation for making their own food. And these help in recycling nutrients from the soil like nitrogen, phosphorus, iron, and sulfur. So moving on with this, so talking about some of the heterotrophic bacteria. So heterotrophic bacteria are the most abundant in nature. Yeah, these are found everywhere in nature. Also, the majority are important decomposers. So these heterotrophic bacteria helps in decomposing the soil better. Also, these help in the formation of humus or by degrading the soil matter, and many of them have a significant impact on human affairs. Also, they are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots. So these are some of the functions of heterotrophic bacteria. Also, some are pathogens causing damage to human beings, crops, farm animals, and pets. Also, cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. So these are beneficial at some point. Also, these are detrimental at some other point. So these act as both. These can be beneficial as can cause diseases. So these are some of the heterotrophic bacteria. Also, I've discussed about nitrogen fixation in one of my videos, so you may check them out. So moving on with the Kingdom Monera. So bacterials are of four categories I discussed previously. So these are basically spherical, coccus or cocci, the rod-shaped bacillus or bacilli, the comma-shaped vibrium or vibrio, and the last one is the spiral spirillum or the spirilla. So these are the four pictures of the four types of bacteria under Kingdom Monera. So these are some more pictures of them, some of the different forms of bacteria. So you may just check them out. Moving on with this. So bacteria can be classified variously. So the bacteria can be classified broadly into three types: Archaebacteria, Eubacteria, and Mycoplasma. So these can be autotrophic as well as heterotrophic. So talking about these can be autotrophic as well as heterotrophic. Under autotrophic, these can be photosynthetic autotrophs or can be chemosynthetic autotrophs. So we just discussed about chemosynthetic autotrophs. So what are they? Also, we'll discuss about photosynthetic autotrophs in the coming videos. So this is the basic layout of the bacterial classification. So these can be of three types: Archaea, Eubacteria, and Mycoplasmas. Talk about them. So talking about the Archaebacteria, so these are extremophiles and live in most of the harsh habitats such as extreme salty areas, which are known as halophiles, hot springs known as thermoacidophiles, and these can also survive in marshy areas or methanogens. So Archaebacteria are the ones which can survive in extreme conditions, and these are the three types of Archaebacteria which can be found in extreme salty areas, hot springs, and marshy areas. Also, Archaebacteria lack peptidoglycan, and these have a different cell wall structure that enables them to survive in extreme conditions. So these lack peptidoglycan and have different cell wall structure that help them to survive in different conditions. Also, some of the examples of Archaebacteria are methanogens are present in the guts of several ruminant animals. So methanogens which are present in marsh areas are present in the gut of several animals. So moving on with this, so talking about some of the eubacteria, or which are also known as true bacteria. So the other name of eubacteria is true bacteria. Also, these are characterized by presence of rigid cell wall, and which motile, some of them have motile, which are known as flagellum. All right. So these have a cell wall, whereas the Archaebacteria do not have a cell wall, and if they are motile, they also contain a flagella. So the cyanobacteria, also referred to as a blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. So some of the eubacteria can be cyanobacteria, also known as blue-green algae. Also cyanobacteria are unicellular, colonial, and filamentous. Can be marine or terrestrial algae. Also, the colonies are generally surrounded by gelatinous shield, and they often form blooms in polluted water bodies. So it also lead to eutrophication, which causes depletion of oxygen in lakes and ponds. So these are some of the bacteria or cyanobacteria that are the essential causes of eutrophication. Also, some of the organisms can fix atmospheric nitrogen in specialized cells called heterocytes, which can be nostoc and anabaena. So these are some of the examples that you may like to remember of eubacteria. So, and some of these functions are very important for knowing eubacteria. Moving on with this, we're talking about the mycoplasmas. So these completely lack a cell wall, so they do not have cell wall, just like peptidoglycan. So they do not have cell wall. These are the smallest living cells. Alright, so these are the smallest living cells, and many mycoplasma are pathogenic in animals and plants. So these are pathogenic in nature. Also, their locations is they are neutral soils or decaying plant material. Right, so these are neutral soils and also decay plant material. So moving on with some of these. So talking about the bacterial cell wall. So these have a layer of a couple of layers of cell membrane. So this is the plasma membrane, as you can see, and on top of that we have the peptidoglycan. Alright, so these are made up of lipotecoic acid and teichoic acid. So these are some of the things that are made of it. Also, these are this is the gram positive envelope, this is the gram negative envelope. So the gram negative envelope lacks the peptidoglycan, whereas gram positive has the peptidoglycan layer, as you can see, whereas gram negative do not have the peptidoglycan layer. So this is a small difference between the two. So talking about some of the important soil bacteria, the most commonly associated bacteria form soil, which are Archaebacteria, Bacillus, Pseudomonas, and Myxobacteria. So these four are common isolated bacteria from soil. Alright, so we'll talk about them. Some of them are Archaebacteria, Bacillus, Pseudomonas, and Myxobacteria, as told. Also, several bacterial species involved in biogeochemical cycles. Also, desulfuromonas species are most prevalent in anoxy sediments. So these are some of the, so for anoxy sediments we have desulfuromonas species. We'll discuss about them as well. So moving on with this, so some of the important soil bacteria, as you can see, so depending on the population, these are classified. So this is a table depending on the population. So the bacteria are the ones which are mostly present in a soil. Then comes Actinomycetes, then we have Filamentous hyphae, we have yeast, we have algae and cyanobacteria, then we have protozoans. Alright, so this is a population table of all of the organisms present in a soil. So talking about the filamentous prokaryotes, Actinomycetes. So we just studied that after bacteria, the second most organism that occurs mostly in the soil are the Actinomycetes. So let's just go through the Actinomycetes and what it says about, because it's a soil bacteria, because it's a soil organism, so we need to get some knowledge about it. So talking about Actinomycetes, these are prokaryotes which look like fungi. So these are prokaryotes in nature and which are basically look like fungi. These grow as filamentous mycelia and form pores. Also, these produce a chemical compound known as geosmins that gives earthy flavor and aroma. So this is a very important point. So these produce a chemical compound which is known as geosmins. Alright, and these some of them are pathogenic nature and the others are saprophytic in nature. Also, these can degrade complex organics such as cellulose, lignin, chitin and proteins. So these help in the degradation of complex organics as well, and these are the prime organisms for antibiotic producers. Also, filamentous bacteria in the genera Coniophora and Streptomyces are the ones which come under Actinomycetes. So this is the kingdom fungi which I'll be covering in the next video. So stay tuned and thank you for watching this video.